It's a pleasure for me to introduce Béatrice Gulbis and Delphine Martiny, who are our next speakers. Béatrice Gulbis is a medical director of the LHUB-ULB, and Delphine Martiny, as clinical microbiologist in charge of the coordination of the multidisciplinary COVID-19 unit, did an incredible job in the first wave of the pandemic by making possible to provide the essential laboratory tests needed to face such stress. It is why I would like to take this opportunity to warmly thank both of you for your daily involvement during this first wave period. Now, Béatrice and Delphine, I give you the floor to explain how you adapted the lab during the pandemic. Thanks again for your involvement in such crazy period.
Thank you very much, Olivier. Good afternoon, everyone. It's our pleasure to share our experience with you today. So, clinical labs play undoubtedly a crucial role in the management and following of the sanitary crisis. At first, of course, by validating and offering reliable diagnostic tools, but also by taking part in the overall surveillance of the cases. Clinical labs occupy therefore a key position between hospitals and clinicians and public health authorities. A good communication between those actors is therefore mandatory, and there are, of course, many actors when we speak about a consolidated lab. First, a few words about the early beginnings in our lab. So, end of last year, a cluster of cases of pneumonia of unknown origin was reported in the area of Wuhan in China. Cases appeared to be, according to the media and health authorities, well contained. In the early 2020, our lab has to face a previously unknown situation with the management of a patient suffering from Argentine hemorrhagic fever. The patient was hospitalized for about 2 months. This case was, as you can imagine, rather challenging for us. First, all residual samples for this patient were traced and destroyed. Then, all lab investigations were conducted in a Biosafety Level 3 lab with enhanced individual protective equipment. All contact cases were finally traced, and hopefully no secondary case was identified among healthcare and lab workers. This experience helped us to improve biosafety procedures, but also highlighted the needs for a global communication inside the entire lab, and also for close contact with our clinicians to ensure the best care for the patient in optimal safety conditions. The patient was still hospitalized when the first COVID patient was admitted at the CHU Saint-Pierre. If you remember, the patient was asymptomatic and fast discharged after the quarantine period. So it appeared like a period of respite for the lab, but a short respite because fast COVID cases increased exponentially and we had to react. This is how the Lab COVID Coordination Unit was born in March 2020. This unit brings together the lab direction, heads of all medical services, 3 microbiologists, each one responsible for one of our activity centers, and all heads of support services, including secretariat and communication, IT, purchasing, quality, and peri-analytics. Daily meetings were organized to treat all relevant topics linked to the crisis, such as, of course, diagnosis, but also the management of the equipment, the communication with the teams and media, the changing guidelines, among others. All decisions and information were transmitted to the teams through memos, internal newsletter in both French and Dutch, also through short face-to-face meetings, but you can imagine that they were rather limited because of sanitary precautions. Actually, we moved from dealing with the samples of a single highly infected patient in January to an entire population susceptible to provide many infected samples and to contaminate our teams in March. Our first priority was thus the biosafety. Several measures were rapidly taken to ensure the protection of lab workers. Of course, we must try to encourage standard precaution measures, such as hand hygiene, wearing protective clothing adequately, avoiding the use of smartphones in the lab, all good lab practices that are well known, but unfortunately, I must say, not always fully respected. On the basis of a risk assessment evaluated in all lab departments, we recommended additional precaution measures based both on the nature of the sample and the analytical step and handling. Biosafety cabinets were dedicated to the decontamination of all stool and respiratory samples sent to the several dispatching centers of our lab. In agreement with our clinical partners, the request for several risky and less urgent analyses were also reduced. Finally, we also solicited contribution of the cleaning services for an enhanced cleaning of all contact points, such as doorknobs, power switches, and that was true for all our lab sites. Lab workers were also trained to contribute themselves to the cleaning of their own lab environment by the Brigade de la Vette, which translates as the Dishcloth Squad, an initiative by medical teams of one of our hospital partners. This table summarizes the measures taken in the different lab services according to the risk assessment. In a few words, no additional precautions are taken for blood or urine samples, but biosafety measures are enhanced for both respiratory and stool specimens. These specimens, including fluids and also screening rectal swabs, need to be handled in biosafety cabinet with a laminar flow, where for practical reasons it's not possible. We use a chemical hood with FFP2 masks, and in case no hood is available and there is a risk of aerosolization, lab workers are also provided with glasses. In the same way that medical fields, we unfortunately faced shortages in IPE. Several measures were also taken to avoid the misuse of these equipments and to ensure the accessibility to adequate IPE when necessary. The distribution of FFP2 and surgical masks was controlled by team leaders or by the Secretariat on all activity centers, and all FFP2 masks were rapidly submitted to a sterilization procedure validated by the infection control and pharmacy teams of CHU Saint-Pierre, so they were reusable. Limited numbers of SHA were distributed in the labs and stocks were under surveillance. A special team composed by the purchasing department and volunteers was also dedicated to the search of IPE alternatives. This team worked in close collaboration with and sometimes connected the pharmacy departments of our 5 hospital partners. To answer patients' need, the microbiology lab validated and implemented in routine several COVID diagnostic tests, beginning of course by the PCR when the National Reference Center was overwhelmed, and following by antigenic and serological assays. The experience of our virology team also allowed for the recognition of the virus in samples of patients not suspected to be COVID positive. In addition to this deployment, the microbiology lab worked hard to ensure, as far as possible, a PCR result within 24 hours and helped the clinicians by providing tools for differential diagnosis at the emergency room and rapid COVID PCR tool for particular situations such as transplantation. In the diagnosis as well, we faced shortages of reactants for the PCR, shortages of plastic disposable, but above all, when reactants were under control, shortages of sample collection equipment. In this struggle, our teams worked in close collaboration with engineers from the Free University of Brussels and the Erasmus Hospital to validate 3D printed swabs. The lab also reactivated an ancestral recipe for the preparation in large volumes of a homemade viral transport medium that was rapidly offered to our 5 hospital partners. The development of new analysis was not observed only in the microbiology department, and both immunohematology and clinical chemistry services were also deeply involved in the war against the SARS-CoV-2. As previously explained today, several biomarkers were made available for the evaluation of the disease severity, the prevention of complications, or the therapeutic drug monitoring. This slide illustrates the complete panel of analysis that was finally made available for the management of COVID-19 patients in our hospital partners, but also in other institutions. I give now the floor to Béatrice.
Thank you, Delphine. So, of course, there was an evolution in the clinical activity. As you know, there was a huge drop on the activity and mainly on the ambulatory activity. And in the same time, so we had to allocate the staff to other sectors in the lab, but also to protect them, not only internally, but to be sure that we have enough staff all the time, all during the period of infection, to be sure that we could give the services that the clinician and of course the patients waited for. In the same times, we had probably to clone the sector of biological molecular biology, because you see that in March we had a certain amount of regular molecular biology in microbiology to test for, and then we had the adding of the COVID-19 tests. But nowadays we have the sum of both the regular test and also the COVID test. And it is amazing to know that sometimes several labs, to be sure that they will be able to test for COVID-19, they send us routine molecular biological tests in microbiology. We had also to adapt to the demand, but it was not always easy. So we tried to find other ways to test the patient. And for example, the antigen testing was some of these issues, but it was all sometimes too late. We were too late, and I think that Peter Vermeij explained something of these late issues related to the recommendation, the validation, and everything that is very complex at the federal level. We had to adapt to the demand, and how did we did it? We used different instruments from different companies to be sure that we don't had the problem with the shortage of delivery of the reagents. Then we added different models within the same company. We, of course, added the shift of the personnel and the staff, but it was very hard for them because they were not used to work during even shift and during the weekends. And you know that you have here in the black line the capacity that has been asked by the federal government, and it was based on what? On the symptomatic patient to be tested. They just count that each day we will have 50,000 contact patients with symptoms or signs of upper respiratory infection. And then we will try to gain the maximum capacity with all those different issues. We had also to adapt with the recommendations, and of course it was quite different, and Marie-Gilbert give you some of the answer. We test the symptomatic patient and very severe patient or moderate patient, but never the asymptomatic. But during July and August, we saw that we could have stock to give some peace to all our personnel, but it was impossible because in the hospital they wanted to test all the patients that enter the hospital, like for a pre-operative purpose, and then we had to test all the individuals coming back from the orange zone or red zone, and so it's very, very difficult. And you can see the difference of the all different individuals that we test today. We had also to help several partners. In the beginning, I think that, of course, we helped, as other labs, the National Reference Center, which was really overloaded. But then we helped also other hospitals through our partner of clinical labs. Then we helped all around different collectivities, and like homeless, for example. But we help also for scientific studies, which is one of our mission, and we didn't want to avoid it, of course. And we had also the federal platform because the triage center were overflowed or not ready in April. What was our duty also? It was to give information to all our partners, the clinician, the stakeholders, and so on. So we gave them statistics, but also all the time, every day in the beginning, relevant information to be sure that we had a link and that we understand each other. We also give a tool to give the positive cases to all our clinicians on a real-time basis. It was very, very important. And you know that nowadays we have to give our data in the hour after validation to the federal health data. And we participate as the other, of course, to the epidemiological surveillance. We had also to adapt to something very tricky, which is all the systematic tool, informatic tool that was implemented for testing and tracing. So we are somewhere here, and you are all in the lab are also there. We had to send the data to the COVID-19 database to be sure that the center for the tracing could work, and the regional health inspector will be informed, and of course to give by Cienciano the epidemiological data. But the information was given in April 30, and the implementation had to be done on May 4. And then to adapt, what can we do? There was, for example, for serology and for SARS-CoV-2, we had a royal decree in the beginning just to be sure that the autotests were prohibited. But then in April 3, we get a directive telling us that there will be a reimbursement for serology, but that the patient billing was prohibited, and it is still prohibited. We had indication of how to use the serological tests for when can we get the reimbursement, but then finally the real decree for the reimbursement was in the middle of May, and an update only in June. So it was really a very difficult period for us, because a clinician and a patient didn't understand why we couldn't give them the serology, while we were, of course, ready since several weeks, as Carlota Montesinos and Olivier van den Berg told you. Fortunately, we had very nice information every week from the National Reference Center, and we thank them for that. But of course, we were involved in different federal meetings, and this is also very important to get all the information that we can get to be sure that we prevent more than to adapt in a rush all our system. So what are the lessons that I learned? Can we get them to prepare the near future? But we are in the future, I think. So the teams have worked in rush and sometimes in fear. But we have seen a very, very great solidarity. The collaboration was really very good, so this is good to know. What we've learned also, that whatever the different channel of communication you use, you can't touch each individual, because the way you feel the crisis is probably different from one person to the other. But try to encourage them and congratulate them as much as possible. It's very important. We have also learned that the link between the lab and the hospital is something very important. We have to strengthen it. Of course, since we are a lab working for 5 different hospitals, it's tricky to have those communications, but it improves, and we are happy for that also. We have to know that all the parameters can't be under control. It's impossible, because some decisions are made very above, so we can't know what to do. But try to be creative, have a reactivity, big collaboration, and preparation are some keys of the success. And collaboration is not only inside your lab, but of course also outside the lab. There are political decisions, but they don't have to always drive yours, but they have to be part of them. So what are the key message? Of course, the laboratory has and has a key role, but believe me, we are only a part of the chain, and if all the people, all the citizens in Belgium and in Europe don't follow the rules, we will be really in a mess. And then try to take advantage of your experience and also your mistakes. We learn from our mistakes. We know, all of us know that. So we want to, Delphine and myself, and under the name of the direction of the lab, we really want to thank all our collaborators. Yeah, it was a real solidarity, and it has to be now, today, and tomorrow, unfortunately.
Thank you very much, Beatrice. Thank you very much, Delphine. Just one question, because time is moving. Delphine, if you have to summarize this experience in one word, which word do you choose?
It's a difficult question, but I think I would say collaboration. We need to work all together inside the lab, but also in close collaboration with clinicians, as Béatrice explained, in collaboration with health authorities. So I think collaboration and communication are crucial and will be mandatory for the next step as well.
Thank you.
I think that we've learned a lot inside the lab with different skills, competencies, and it's the same with all our colleagues in the different hospitals and even in the lab with whom we are working with.
Thank you very much.